Hello, students. I'm here to talk to you about AP Biology review with cicadas. I'm going to talk a lot about cicadas, but I'm going to tie it into all the units in this curriculum. So, to start, let's talk about the double periodical cicadas. These are the ones that are coming out this year. Sometimes they're called the Mag's cicada. I think that's pretty excellent. These are going to be the red-eyed ones, not the usual ones. Now, it's a weird year because we are going to get 17-year cicadas, brood 13, and 13-year cicadas, brood 19, that are just happening. The last times these were out in 2007, now it's 2011. We're getting them both this year. You can see that in good old Illinois, there's going to be a little bit of overlap. It's not going to be a huge amount of overlap, so you're not going to be your whole house isn't going to be covered in them, but it's kind of a weird, fun year where we're going to see lots and lots of cicadas. So let's talk about them and how they connect to AP biology. This is a phylogenetic tree. In AP biology, being able to say the words recent common ancestor and node are going to be really, really helpful for you. So anytime one group splits into two different groups, and you could see the Decula group and the Cassini group, they split off about 2.2 million years ago. You can see it at the node right here. These lines are, stopped, but they're actually much longer. But yeah, these ones actually follow this nice 1,000 years ago trend when you could see each of these splitting off from each other. So being able to read something like that, this is, from an actual paper, this life cycle control of 13 and 17-year periodical cicadas, which I just ripped right here and wanted to show you, hey, if you've been paying attention, you can interpret this. There's new words here, but yeah, this is active research right now. Being able to interpret error bars are going to be super important. We talked about it very early in the year in my class, but I want to show you guys that, yeah, this is, again, same paper. They showed, this is a line graph because it's two different numbers, the concentration of this steroid that I'm going to talk about later and length. And they showed, different groups. They show that there is overlap between these groups here because we see overlapping error bars, but we see they're not overlapping between these groups, right? So these are overlapping, these are not overlapping, so these are statistically different when you're interpreting them. Know how to do Hardy-Weinberg. So remember the 5 ways to violate it. Know that this is kind of like a null hypothesis. It's a way to measure how much evolution has actually occurred. Remember the 5 criteria that violate it, or the 5 criteria that actually cause it to be in Hardy-Weinberg, which would be violated in an evolving population. There is no migration, no mutation, no natural selection, no sexual selection, and no genetic drift, so very large population. If you violate any of those, you'll move away from Hardy-Weinberg equilibrium. Note that the best thing to do in Hardy-Weinberg problems is usually figure out that Q squared, which is going to be due to the recessive trait if we're assuming Mendelian genetics, Mendelian dominance. And so, because if you know what causes recessive, then the recessive individuals, again, the frequency, the number out of the total is going to be your Q squared. You can take the square root and you can get Q. And then if you know Q, you could take 1 minus P, or 1 minus Q to get your P of 0.3. And then that opens everything. Remember, 2PQ is the frequency of the heterozygous. P squared is the homozygous dominant. Q squared is the homozygous recessive. If they want just the frequency of one allele, it's going to be P. If it's two, the recessive, it's going to be Q. If it's individuals, you need 2 alleles. If it is just the allele frequency, then you only need 1. So if they gave you this problem, suppose 49% of cicadas, of a cicada population has a recessive trait called flippant wing What percentage would be homozygous dominant? Well, this is how you do it. That'd be 0.49 is q squared. You take the square root and you get 0.7. And then since they want to know homozygous dominant, you need to figure out p. 1 minus 0.7 is 0.3. Okay, now you have both of these. And then percentage would be homozygous dominant. That's two different letters. That's p squared. So you would do 0.3 squared and you get 0.09, or as a percentage, 9%. This is just an example. Moving on to the next unit, we talked about biochemistry. In cicadas, there's this really important ligand called 20E. It looks just like this. Now remember, at each one of these locations, because carbon and hydrogen are so important to biology, they're just not even included in these line drawings. So all of this is mostly carbon and hydrogen. There's a little bit of oxygen here too. And remember, we talked about oxygen being a huge electron hog, so it causes things to be polar. Things that are polar can't get through. Non-polar things can get through a cell membrane. So this paper, a different paper on insects, and this 20E is 20E. Even though it's a lipid, it's pretty big and it can get through, but it gets through easier with facilitated diffusion. Remember the difference between these two. Facilitated diffusion still is going from high concentration to low concentration, but you're using a nice protein channel to make that possible to get across here. And it's likely because of these oxygens here making it more polar. So let's talk about that a little bit more. So hydrophobic molecules like O2 and CO2 and N2, most steroids can get right across the cell membrane. But small polar things, they can't. This is mostly hydrophobic in here, so they can't. So H2O and glycerol and urea, ethanol, these are little guys. They have a hard time getting through. I'd say that 20E might fit here, even though it's mostly steroid. It's kind of between these two. Probably needs a transport protein. Large things definitely cannot get through. Polar or non-polar. So glucose and sucrose, they are going to stay on one side and they need some sort of protein to carry them across. And then ions, because they are so polar and charged, they also can't get across. You know, your sodiums and your calciums and your chlorides can't get across at all. These can. These have the easiest time, and these have a much rougher time over here. So I talked about steroids. Steroids are lipids. That 20E would fall under this category. It just has C, H, and O. If it's a phospholipid, then it's going to have phosphorus as well. It's mostly hydrophobic. Again, all the carbons and hydrogens make it much easier to get across that lipid membrane. The monomer is going to be fatty acids and glycerol. Carbohydrates, notice I have a capital O here and just a lowercase o here because carbohydrates have a lot more oxygen. That's where the O comes from in carbohydrates. They are not hydrophobic. They have too much oxygen. And glucose and fructose, other oses, are going to make up your carbohydrate chains. Proteins, they have these elements. I like to think of them as honks because they have sulfur. They don't have phosphorus, but they have nitrogen. Too hydrophobic, too much nitrogen and oxygen to get across that membrane, and amino acids make them up. We're going to talk about proteins more. And nucleic acids, I remember with FONC, they are also not hydrophobic. They can't get across that cell membrane because they have too much nitrogen, phosphorus, and oxygen, and they're made up of nucleotides. Now, proteins, again, made up of amino acids, they have other levels of structure as well. This is the receptor that binds 20E that I talked about before in cicadas. Notice you see lots and lots of these structures here. These are the alpha helices, that secondary level of structure. You also get to see some beta pleated sheet, but it's a lot of alpha helices here that look just like this, not DNA double helixes. These are alpha helices. And they are part of that secondary level which is due to hydrogen bonding between amine groups and carboxyl groups on different amino acids in that chain. Um, the primary level of structure, that's just the chain of amino acids that's got strong peptide bonds, which are covalent bonds between them, um, are again made during dehydration synthesis reactions. Secondary, again, is that hydrogen bond. These are the weakest because hydrogen bonds are so weak. Tertiary is the next level up. It has these weak hydrogen bonds, but it also has strong disulfide bonds and ionic bonds, and some other weak hydrophobic interactions between the R groups of different amino acids. And quaternary follows the same rules, but if you have more than one polypeptide chain, like in hemoglobin, then you'd have quaternary structure that uses R groups or side chains and cares about these interactions here. Now, moving on to the next guy, we've talked about biochemistry. This is just a overview. I'm not covering everything because I want to get through stuff a little faster, but and tie it into cicadas because I think it's fun. Um, that 20E is a lipid. Lipids are made in the smooth endoplasmic reticulum, which remember is different than the rough endoplasmic reticulum, which has these ribosomes. It's kind of these channels where proteins are made on those ribosomes. We've got our nucleus here too. That's going to store the DNA. That's where the nucleoluses are that make the ribosomes. Here, in general, I don't show them all, but you should know your organelles. It's definitely worth reviewing, but I thought this would be a nice little interesting tie-in. Again, this is all going on in cicadas. Um, Cell signaling, that 20E, where you may be thinking, why do I keep talking about it? Well, it's one of the important proteins for when cicadas do their metamorphosis. That metamorphosis, they're going to be coming out of the ground. They're going to be making that wild noise, but they need to go from their nymph stage into their adult stage. And this signaling here is part of that. So 20E, it'll bind to a G protein couple receptor, a GPCR. This is going to be the stimulus. When the right temperatures are hit and they're ready to move out of the ground and up. That's the stimulus. The 20E is going to be in the system of the cicada, and it's going to bind to the GPCR. That's the reception, and then you get the transduction. Remember, arrows usually mean that these things are being activated. And these little blockers mean that this is causing inhibition. So we can see when 20E binds to this receptor here, it's going to stimulate GAQ or PLC, which is going to stimulate this ORA1 and calcium can get in. And then we see PKC gets activated too, and that is going to block this because you've got enough signal. And then we're going to see some 20E binding to receptors in here, like I showed you earlier. And then you get transcription. It turns on this gene, and then you get this overall response. Again, this is just a signal transduction pathway. Make sure you can read. You won't need to know what all these things are, but read and know what's getting turned on, what is maybe getting turned off in these mechanisms here. So all of this stuff is going to take lots and lots of energy, and cicadas. Let me see if I can get this to work. They make this noise. It takes lots and lots of energy to make this noise. I'll maybe play a little more. Lots and lots of energy to make that noise. So you're going to need to get that energy from somewhere. And the cicadas, they're getting it from sugars. They're getting in the xylem of trees, especially underground, but eventually in leaves. And that is going to provide them glucose, which they are going to turn into pyruvate, and ATP and NADH, which they're going to use if they've got oxygen available. This pyruvate gets oxidized. Remember, oxidation is when you're losing electrons into acetyl CoA, which is going to go into the citric acid cycle, which we've also called the Krebs cycle. More NADH is getting made, and you get a little CO2 as a byproduct. The citric acid cycle If you have NAD plus and FAD available, it's going to make NADH and FADH2, a little bit of ATP or GTP really, and a little CO2, which is going to feed into oxidative phosphorylation or the electron transport chain. All of these NADHs and FADH2s are going to give up their electrons. That provides the energy to pump these H pluses across here and eventually make that really, really useful ATP. Now, I said you need oxygen for aerobic respiration. That's because these electrons have to go somewhere. If you don't make this, or if you don't have oxygen here, you can't make water. You can't take these electrons, and this whole NADH process breaks down. So all you can do is glycolysis in the cytosol. Don't forget, this is also these parts in the mitochondrial matrix, and this is the inner membrane of the mitochondria. It takes lots of energy. All right. Know about photosynthesis too and how it interplays with respiration. Note, I just talked about all the things in respiration. I won't belabor that point. But byproducts of respiration are H2O, which is needed in photosynthesis, and CO2, which is made in the Krebs cycle and that pyruvate oxidation, which is needed for the Calvin cycle over here, the carbon fixation by Rubisco to make PGAL or G3P and eventually glucose, which will feed into respiration. Those light dependent reactions, they require water as electron donor and then light in order to excite those electrons to make ATP and NADPH, which, like NADH and ATP, is like another electron carrier into the Calvin cycle to reduce some stuff so that you can make PGAL. This ATP is not the main driver of energy. It's just providing the energy needed for carbon fixation and making this, because in plant cells, you're also going to be doing those plant cells that the cicada is going to eat. You're going to do respiration as well. In the cicada, it's got all sorts of digestive enzymes while it's eating. And this is how those enzymes work. Don't forget, they are going to they're going to lower that activation energy by holding those reactants so they can easily become products. And that makes the reaction much more likely to occur. So this is nice cellular respiration here. If you didn't have all the enzymes involved in cellular respiration, it'd be, glucose would break down into with oxygen into CO2 and H2O, but it'd be much, much less likely because the activation energy would be so much higher. You need a lot more energy put in. And let's say you were trying to identify the difference between these two cicadas. These are the periodical ones, and this is the typical yearly cicada, which are harmless little guys. They might harm your little trees, but they're not going to hurt you. They're herbivores. Well, if you can't see the difference in eyes, that's one of the big differences to see here. If you're colorblind, you would have that difficulty. So let's talk about color blindness and how it ties into genetics. So don't forget, sometimes these are labeled, sometimes these are not. The squares are the males and circles are the females in pedigrees. We can see the males have color blindness in this pedigree a whole lot more frequently than the females do, strongly suggesting that it is a sex-linked trait, and color blindness is. This is a nice little Punnett square showing that if you have a male that has the blindness, right? Color blindness, or non-color blindness, I guess would be the female that is not colorblind. You would see that if the female's got one of the wild type or the normal X chromosomes with color blindness on it, that would just lead to carrier daughters that don't have it, with this lowercase b being the recessive. Sometimes you see it up here. But yeah, all of these would be carriers, meaning they could pass it on to their children because they have the one normal type. But all the boys would end up being colorblind of the children because they have this Y chromosome that doesn't have anything to cancel out the recessive trait here, which is why it would show up so much more frequently, the actual trait in these sex-linked genes here. And back to our good friend cicada, they could ask good ecology questions, know some of these terms. Any sort of plant that does photosynthesis is going to be a primary producer of energy. The primary consumers are the things that are going to eat those plants, like cicadas or caterpillars. Secondary consumers eat the primary consumers. And then tertiary consumers, which are oftentimes apex predators or things at the top of the food web, they're going to eat lots of different things. They can sometimes be keystone species because if you lose your snakes, then you get too large a caterpillar species and too large a mice and too large a shrews. And because of that, then they might eat too much of the producers, and it might be really, really unhealthy for the ecosystem. And sometimes they might ask you about population calculations. Say you wanted to know what the maximum growth rate would be for a cicada population, and they might give you it has a carrying capacity, or they might not. If you're given a carrying capacity, you should think this, logistic growth, or if you see this, you should think there is a carrying capacity, where K is the maximum level. It might bounce about that carrying capacity, but that still means that it's the carrying capacity because you're not, growing exponentially like here. But if you've got ideal conditions, you've got no disease, you've got no predators, you might have exponential growth like this kind of J curve versus this S curve here. Um, and again, if they're asking for the maximum or their growth or the growth rate, they're looking for you to work with the R and solve for the R. N is the overall population, and DNDT is how much it changes in a year. So I'm excited about the cicadas. I hope you're excited about the cicadas. You're excited to do well on this AP biology exam and then go some cicadas. Go read about these wonderful critters. They're pretty neat. A lot of people are worried about them, but I think it's just going to be a really cool thing that's happening. That's pretty rare. So go out and enjoy this and enjoy AP Bio. Thanks for listening, and I wish you well, and can't wait to see y'all on the other side of this exam.